Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of the show, bringing another really awesome guest for you today. Today on the show, we have the honor of being joined by Dr. Ben Hwang, who is the Chairman and Chief Executive Officer of Profusa, which is a pioneering bioengineering company. They're developing tissue-integrated biosensors ultimately designed to continuously measure key chemical markers in the body, and their mission is ultimately to make our body's chemistry as accessible as things like heart rate or step count, ultimately transforming personal and societal health through these real-time molecular insight tools. Dr. Hwang has spent his career at the really interesting intersection of life science tools, of digital systems, global markets. He was an undergraduate research fellow in the lab of systems biology pioneer Leroy Hood at Caltech, witnessing firsthand ultimately how some of these transformational technologies ultimately will help redefine biology. He went on later to hold senior leadership roles at Life Technologies, now part of Thermo Fisher Scientific, including president of Asia Pacific, as well as head of the qPCR division, ultimately helping to scale molecular biology platforms globally during that really pivotal area of both genomics and diagnostics. And earlier in his career, Dr. Hwang worked at McKinsey. He interestingly began his career at the LA Dodgers, a really interesting trajectory ultimately in his career there. Did his master's as well as his PhD in biology from Johns Hopkins. And a lot to get into today. Really excited to have him. Dr. Ben Hwang, welcome to the show.
Thank you so much, Ira. Appreciate the time, appreciate your interest. It's really nice to talk to you and to your audience.
Yeah, no, I'm looking forward to getting into to a lot of cool topics today. But let's start off, though. I'd love to start off with the journey. You started as a bat boy with the LA Dodgers. You end up in Leroy Hood's lab at the time where he's messing around trying to create the first DNA sequencers. And then you end up on the East Coast over here at Hopkins studying sodium potassium subunit assembly and transmembrane proteins and all that bit for your PhD. Take us back a little bit. I want to hear the story before the story here.
Yeah, Ira, do you want the long story or the short story?
Yeah, the long one's cool. I like
it. Yeah. So, it's a story that I've had to explain a few times when I'm applying for jobs and applying for schools. When I was young, I got really lucky. I'm come from an immigrant family. I'm a first generation American from Taiwan. And during a summer, I was volunteering and working at one of my dad's friend's photo shop in Los Angeles. Saw a bunch of Dodgers photographs around, and I asked him, "Hey, where'd you get these? Negatives." And this is back when film was in vogue. And he said, "Oh yeah, the Dodgers photographer used to own this camera shop." And that led to well, can you give me an autograph ball? He said, "Sure." That led me to, well, if you give me an autograph ball, can you give me a job? He said, "Let me ask." And that led to one day tryout to be the bat boy for the LA Dodgers. And I didn't play baseball. I was horrible at it. And I didn't know that being a bat boy, you had to throw a baseball, know how to throw a baseball and play catch with the players. So I had to really learn how to throw a baseball. And that one day turned into 3 days, 3 days turned into a homestand turned into wanting to work the rest of the month. And rest of the month turned into 6 years at Dodger Stadium. And after that happened, actually during that time, I had actually dropped out of high school for just a variety of reasons. And so now I found myself as a 23-year-old at Dodger Stadium, and by this time I was working in the front office from the clubhouse, and being a high school dropout, and I had a decision to make. Do I want to go back to school, or do I want to literally have the best job that any kid could ever ask for if you're a baseball fan? And I thought, okay, let's have a longer term view on life, and let's go back to school. And so went back to school because I didn't have a high school diploma. I had to go to community college, and I went to Pasadena City College. And during that summer, I needed a job. And at the time, I was pre-med, and I started looking for a job. And one of the paths to finding that job was walking 2 blocks to Caltech and knocking on doors at the Beckman Institute, trying to find a position where I could get closer to the sciences, because at the time I was had aspirations of being a physician. And I knew so little. It's almost embarrassing for me to admit this at this point, because I like to think I'm a pseudo expert in the life sciences. But back then I knew so little about how academics work that I kept knocking on doors and say, hey, do you guys need somebody to sweep the floors or wash test tubes and such? And the answer is, of course, no, 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 no. And finally, somebody, I guess was a graduate student, was kind enough to say, you should, like, don't knock on doors. Nobody's going to say yes to you knocking on doors. Go to the department office, get a catalog of the work that's being done by everybody in the department, find a lab you're interested in, and then go ask that professor if you could have a job in that lab. So I did as he suggested, went to the department, got a printed catalog. And of course, I could pronounce the words, but I didn't understand anything because of the jargon. So my naive way of thinking about this is, well, I don't know anything about science. I had just taken a general chemistry and general biology class at a community college. I'm knocking on doors and wanting to work in the lab of one of the most prestigious scientific institutions in the world. I'm going to stick out like a sore thumb. So the lab that I'm going to target is the lab that had the longest list of postdocs, research associates, and graduate students. Because I figure if I go to a lab with 3 people and I stick out like a sore thumb, everybody's going to know. If I go to a lab with 200 people, nobody's going to know I stick out like a sore thumb, and I don't know what I'm doing. And that lab happened to be Leroy Hood. He had more than 200 people in his lab at the time. And little did I know that was completely the opposite strategy, right? The strategy is you want the small lab, because usually that's a assistant professor just getting started and always looking for eager hands. So I knocked on the door of Dr. Hood's lab, and the lab manager, Dr. Joan Kabori, I still remember her well. She laughed. She said, well, chuckled, right? And wasn't laughing at me. She's like, well, okay, it's nice of you to actually come looking for a job, but we don't have jobs like that, right? We have people who are scrubbing toilets, people who are going to wash test tubes already. But we do have this opportunity for a summer fellowship geared to undergraduate students. If you want to apply for that, here's an application form. And I said, are you sure? Because there's a pile of dishes right now in the sink that I could see in the lab. I'm happy to walk over and wash the dishes right now. No, I'm sure. We got people to do that. Take this application. I took the application. Sorry, this story is getting a little long, but I took the application, went back to Pasadena City College, found a typewriter in the library, and started typing it out. And the application, when I was done, it was empty. And the reason it was empty is you could imagine what an application for an undergraduate student to try to get a summer undergraduate research fellowship is like for Leroy Hood at Caltech. Mm. It is typical, right? Name, address, zero rank, but it's also, okay, what classes have you taken? What research have you done? How many papers have you published? And what is the area of interest in the work that we're actually doing? And of course, as an undergraduate with half a semester under my belt at Pasadena City College, it's my name, address, Pasadena City College, General Chemistry 1, General Biology 1A, and that's it. No publication. So that application was completely blank. I walked that back over to Joan Kabori, Dr. Kabori, probably within a half an hour, because it didn't take me that long to finish it. She thought I had a question on the application, and I said, Dr. Kabori, here's the application. It's done, but I'm really embarrassed. I hope you don't mind. I'm really embarrassed because I just don't have anything to put on here. But I'm incredibly interested in the sciences. I'm a hard worker. I like to think I'm reasonably smart. And if I don't get this fellowship, my offer still stands. I'm happy to come in and just wash test tubes because I want to just be around this environment and understand what I could contribute here and what I could learn. She chuckled again, lovely lady. Don't worry about it. Well, turned out I got one of the spots in the fellowship and it changed my life. Now, I was right. The other 9 fellows were from institutions like Harvard, Brown, MIT, Stanford, who are juniors and seniors with long publication records. And I spent that summer learning how to pour agarose gels and run PCR reactions, but I learned a ton. And that turned into Dr. Hood, kind enough at the end of that experience, asked what I wanted to do. And he made an introduction to a professor at Johns Hopkins. Mm-hmm. I believe one of his roommates when he got his MD at Hopkins. He got his PhD at Caltech. Dr. Hood did an MD at Hopkins. And when he was there, I think Dr. Fambro, who turned out to be my PhD dissertation PI. So I flew to Baltimore, met him, and he said, well, Lee seems to really like you. So if you want to apply to Hopkins, I'm happy to write you a letter. And I got in. And that's how I ended up at Johns Hopkins. Not Johns Hopkins.
No, that's awesome. And it's just one other place I want to ask you. I just got to ask you about it while I have you. Then we'll get into Profusa and
sure.
talk about this.
Sure.
But you're also, after that, you're, to me, as I mentioned in bio, you get to life sciences and you're leading, this company at a really important part of sort of the genomics revolution. We had Craig Venter on last year, and clearly you're no doubt providing not just sequencing tools, but cheaper, faster, smaller tools over time. And obviously we'll make some comparisons here to biosensing in a little bit, but again, say just a couple of words about that era, because I find that really fascinating as well, what you probably saw during that time.
So I'll say this. When I was during that summer research fellowship at Caltech, there were 2 things that was sort of seminal in life sciences. I may not have appreciated it back then, but now that I've been in the industry for 20 plus years, looking back on it, there were 2 seminal events, right? That for me, anyways, at the time. One was the CFTR was just sequenced. So Francis Collins had just sequenced and found the first major mutation, Delta F508, in the CFTR that caused cystic fibrosis. And that was like major news, tons of work to just find this one mutation. And cover of Science, very well covered, incredibly important discovery. And then the second thing that I remember was right outside of the lab that I was working in. I was working with one of Dr. Hood's associates, research associates, Debbie Nickerson. Right outside of the lab, there was a big poster that Dr. Hood's lab had just presented at some conference somewhere and brought it back. And it was the schematic of the development of the capillary electrophoresis DNA sequencing system, right? The 4-color, 1 capillary tube. And, at the time, like I said, I may not have appreciated how important that was. But in retrospect, once I joined, so I got my PhD, I joined McKinsey, cut my teeth and learned a lot about biosciences, and basically came home in the life sciences industry and joined Life Technologies. And Life Technology, to come first full circle, when I joined Life Technology, the company was actually called Invitrogen, and Invitrogen merged with Applied Biosystems to become Life Technologies. And Applied Biosystem happened to be the company that spun out of Lee Hood's lab for that electrophoresis sequencing machine. And it dawned on me somewhere around that time that technology fundamentally could change the world. It sounds like such a cliche, Ira, and I hate using it a lot, but technology fundamentally can. Memory chips, silicon boards, integrated circuits, internal combustion engines. In life sciences, I would argue that automated DNA sequencing took the solution in trying to solve the human genome from an impossibility in our lifetime into something that we do routinely. And it all started with this brilliant insight to say, how can we do this a little more efficiently? And then it allowed Craig Venter to actually daisy chain it. And next thing you know, the human genome is actually solved. And it was incredibly exciting to be during that whole genomic rush to get the cost of sequencing as efficiently as possible, because that one machine spawned and inspired so many other multiple approaches. And it spawned so many other, not just physical sequencing approach, but how software and piecing DNA together, shotgun sequencing, et cetera, and created a bunch of efficiencies that really made the cost of genome to come down. And when the cost of genome started coming down, it opened up entire bodies of science that we could not have possibly imagined just 10 years prior to that, right? Now we can't have a conversation. You don't have to talk to a scientist. You talk to anybody, and our fundamental understanding and the latest tools and technologies that have been developed in life sciences have all found their way, for the most part, because some genome or some DNA sequence or some DNA to protein mechanism is understood. And you could all trace it back to this machine that Leroy Hood actually invented. And it's what got me so excited about Profusa and what we're actually doing. Because I think there is a really good parallel analogy there, right? Oh, yeah. As smart as Dr. Hood is, and he's probably one of the smartest guys around, he takes information, put it together, and be able to be visionary. And as visionary as he is, I'll bet you he would be shocked at some of the stuff that's actually happened and how the world is really different, and how life science is really different, because we have the human genome and all these other genomes solved and learn from each other. For us, I actually think that's the case. It's really easy to understand how, if you have continuous biochemistry monitoring that replaces the data today for most biochemistry, which is point-in-time blood draws. Mm-hmm. It's easy to understand how, if you could take a movie as opposed to taking a picture, taking a movie is much superior. And we know that, right? Real-time information on our phone changed commerce, changed advertising, change video surveillance, change security, change how you go from point A to point B. I mean, it's everywhere. It really changed the way we live. And in biology, absolutely would change biology. But I would actually argue that for as valuable as that alone could be in terms of managing chronic diseases like diabetes, COPD, critical ischemia, wound care, where, kidney failure and kidney dialysis, where having continuous monitoring of biochemistry is really important for those chronic diseases, those verticals. I would actually argue that in order for us to actually reach our potential as technologists and as a society, for the vision of digital health, for the vision of truly understand ourselves as individuals, there are incredibly large swaths of the sky that have not been opened up yet, and can only open up if you have this information. Similarly to the automated DNA sequencing story.
Absolutely. And this is a perfect segue into, what you refer to as sort of this measurement gap, because as you're saying, there's as you do with the genome and the ability to routinely do something with regard to biosensing, we sort of have these three categories. There's the stuff that we can measure in real time constantly, so heart rate and certain clinical measurements per hospital setting. Then there's a lot of stuff which we do episodically, which requires using blood draws and lab assays and all that. And then there's an entire blind spot. And this is where tissue oxygenation and lactate and hormones and inflammatory cytokines, there's a lot of things that fall in that sort of invisible basket, which is what Profusa is ultimately going to be bringing alive. Talk a little about this gap, sort of like, how we got here, where we are, and then we'll transition into what the Profusa tool is all about.
Yeah, I think there are three categories of why the gap exists, right? And by the way, these are not criticism. I think it's actually goes to the ingenuity of the giants that came before us, right? One category is I have a really cool way to measure something. How can I apply that to measure the body and give me some insight in healthcare that we haven't gotten before? So if you have a very sensitive motion detector, for example, with some sophisticated algorithm. Then you could count steps with some degree of ease and regularity, right? And so let's go ahead and measure that. And what problem are we solving? Well, we're not solving any diseases, but hey, we could actually align to people's willingness to be healthy. And over a large population, that's got to create some value for the healthcare system. But it starts with the technology itself. Like, it's already developed. What could it measure actually in healthcare? And I think, measuring heart rate came along the same way, right? You have stethoscope where you can actually hear, or a pulse that you could feel. And then clever folks say, hey, if you can actually have a mechanical perturbation that we can actually feel with greater degree of sensitivity, then we could measure heart rate with something that you strap to your wrist. Because if you could feel it with your hands, then you can actually feel it with this piece of equipment. So let's measure heart rate in that way. So I think there's one, which is there's a technology that already exists. How do we actually improve it so that it's easily deployable and people could wear it and happen? Unfortunately, fortunately, those are great technologies. Unfortunately, though, it actually just covers a little sliver of real important healthcare decisions that actually have to be made, right? Somebody's walking 10,000 steps a day. For most people, that's healthy. But if you ask my dad to walk 10,000 steps a day, that request will probably kill him. If you ask Michael Phelps to walk 10,000 steps a day, it probably doesn't do anything because he's already so healthy, right? So it's not very usable. There's a second category, which is everything is done by blood tests. And I think everything that's done by blood tests has historical reason, right? It's a humor that's available. And so there are decades, if not centuries, of us thinking that, hey, there's something easily attainable in terms of liquid in your body, which is the blood. And then these tests are developed off of that, what's inside the blood, and the insight comes from that. And there's some limitations there as well. Some biochemistries are not carried around in the circulatory system. If you have vascular disease, vascular compromise, it may impact actually how you think about blood measurement versus how healthy you actually are. And so there's some limitations there as well. But I think there's a little bit of, historically, there's all the you build scientific knowledge step by step by step. And if everything came before you is coming from this compartment of the body, then it's tough to actually say, well, let's measure something else, right? You just get insight from that compartment. And next thing you know, you have this, if you will, field inertia that makes it very difficult for you actually to change around. And then I think the third part of this measurement modality is actually time. Most of the assays are done because it's a static assay. You have to take something out of the body, and then you develop the assay to actually measure it. And it's very difficult to actually do that without taking a vial of blood or a drop of blood out of somebody's body. And once you actually do that, then it's a point in time measurement. And clearly it's incredibly valuable. Life sciences and healthcare has gone very, very far with those three ways of actually measuring things. But when you think about those three gaps, or those three kind of major ways right now, where actually people thinking about, imaging or taking biochemical measurements, then it's pretty important or obvious to us that there are a few things that you actually want to do to truly have a stepwise change in terms of monitoring people's biochemistry. One is you should be measuring things that physicians care about, or like at least the clinical community cares about. Because it doesn't matter how intuitive a measurement is and how cool and novel it is. Mm-hmm. In healthcare, first do no harm is an oath that all physicians take. Sure. And unless you have the proof behind it, you're not going to get anywhere in terms of the healthcare system, rightfully so. Now, what that means, though, is you have to measure what physicians care about. It also means that you shouldn't try to measure things that are too novel, because any novel measurements will take decades to actually accumulate the clinical evidence to understand what that parameter means, right? So right now, 70 milligrams per deciliter of glucose means something to a clinician. Mm-hmm. But it only means something to a clinician because there have been decades of studies done on what that number means. So you ought to piggyback on that if you want your technology to actually have some meaning. So you want to measure something physicians care about, do so in a way that leverages what the field knows. The second thing actually you ought to do is let's take care of the temporal problem, right? Instead of taking snapshots, can you actually do this in a continuous way? And doing so in a continuous way means that you ought to deploy a technology and device that, you know, the Fitbits of the world and the Dexcoms of the world have actually kind of gotten a hold of well, right? Which is you deploy it and leave it alone. The question is how long can that last, right? So if something's on the market where you can measure something continuously, but you have to change it every 2 weeks or so, that's a little more cumbersome than if you only have to deal with it once every 6 months or once a year. So how long can we make our sensor modality last and still take continuous measurements all the time? And I think that the third parameter is in healthcare, if we really want to serve society and really want to change in a stepwise fashion the cost and the outcome, you got to make whatever technology you make accessible. You can't make it so that, for the most part, it's a current paradigm, which is you have to be sick enough where your next stepwise change in the diagnosis is so expensive that now it's worth it for an insurance company to actually reimburse you, right? So let's take diabetes. Diabetes is a perfect example. There's no controversy that a pre-diabetic patient, or type 2 diabetic patient, or type 1 diabetic patient, they all need and they all should have continuous glucose monitoring data. Only type 1 diabetes patients have it because they're brittle enough, and when something goes wrong, they go to an emergency room, and then that pharmacoeconomic calculation makes it so that, okay, you know, $2,000, $3,000, $4,000 a year, it's worth reimbursing because ultimately it's going to save the system $10,000, $12,000, $13,000 a year. Type 2 pre-diabetes patients, it's not worth it because if I don't do it well, you just get a little sicker, but nothing catastrophic is going to happen. And so we ought to be able to say, if we want to make a stepwise change, to deploy a technology in a field that could benefit as many people as we possibly can. And by doing so, you're really doing individuals, but also society a favor because you bring down this onerous cost of healthcare, change things into prevention rather than reactive treatment. The only way you could do that is and do that quickly, is let's make sure that the data that we have is what the entire healthcare community could deal with, right? Not steps, but sodium level, tissue oxygen level and such. They know what to do at number. Do it in a way where consumers don't mind using it, or a large population of consumers don't mind using it, because it's easy to apply, it doesn't intrude on their lives, and that data is actually rich and robust, but it's easy to acquire. And the third thing is, let's make sure that the technology platform is low enough cost where if you don't have insurance, it's okay. Out-of-pocket payment is actually still something that most people could afford. And if you can't afford it, we'll figure something else out. Because that figure something else out is not a $4,000 a year decision. That figures something else out is a $200 a year decision. And that's a lot easier to actually do. And when you could do that, then I think you fundamentally could create a ton of value and transform the world.
So in figuring this out, and again, I appreciate you setting all that up. And you know, before we get to the different things that you're measuring that people care about, let's talk about what you put together here, because it's extremely elegant, and I really want our audience to understand this. So, tissue integrated biosensor, injectable. It resides in our body's interstitial tissues, which are, you know, the areas between organs and tissue, sort of the extracellular spaces. So this is not a watch, it's not a patch, it's not micro needles that go into the dermis. This is deeper, but fancy hydrogel-based scaffold that sort of has been equated to almost a synthetic extracellular matrix that you're creating, doing all sorts of interesting, and I'll shut up after this, but luminescent reporter chemistry and molecular recognition elements and all sorts of other things that can measure many things at a time. Again, take a little time to walk through this because it's extremely elegant, but I really want the audience to understand all the aspects of what you put together here.
Sure. No, thanks. So let's rewind the clock a little bit, right? I have to say first and foremost that the co-founders in the company, who are incredibly technically savvy, right? This is their idea. None of this is actually kind of licensed in. And so got to give them a ton of credit, right? Dr. Natalie Wisniewski, Dr. Kirsten Helton, Dr. Kristen Hilton and Bill McMillan, Sawyer Gamzee and such. Give a shout out to them. What they realized though is a couple of things. One, we build off of the shoulders of giants, as we mentioned before, and this whole idea of continuous biochemistry monitoring for diabetes management and CGMs, continuous glucose monitors, idea has been around for a while. The issue has always been that if you put a sensor inside the body or any sensing element inside the body, that whether it's a little tiny needle, whether it's a bunch of microneedles, whether it's a big hockey puck, it doesn't matter what you put in. The body is so exquisitely good at determining what belongs inside the body versus what doesn't belong inside the body. It mounts what's called a foreign body response against that. And that foreign body response is an immunological response. And if immunological response doesn't do it, the body is elegant in depositing scar tissue, collagen around that foreign element so that that foreign element cannot go around and hurt the body. So any of us who's ever had a splinter understands this phenomena. It doesn't matter how small the splinter is. It'll get pussy. You may not even know that the splinter got in. And then at some point it's like, oh, wow, there's my fingertip hurt. And you go, oh, there's a little splinter in there. And then most of the time it gets expelled out. But if it doesn't get expelled out, you're going to live with a bump under your fingertip for the rest of your life. And that bump is just a scar tissue around it, right? So that foreign body response is really difficult to overcome. And a lot of people have tried to, the giants that come before us have tried to overcome it by saying, well, let's not put something inside. Can we draw fluids out of the body and measure it? So microneedles, kind of a semi-approach. People try lacrimal fluid right in the eye, try to do it with sweat and such. And many of those devices actually have failed just because those things are really so difficult to do. And the fluids may not be representative of what's happening inside the body by the time you get it out. And then third way of people doing it is absolutely completely non-invasive. Shoot energy waves, some kind of energy wave through the body and trying to look at the energy wave absorption signature of glucose and see if you can actually tease it out. And it turns out that's almost an impossibility for reasons that probably requires another 2-hour conversation. But to do that, it's almost an impossibility. So when the company sat down and said, hey, is there a company to start? They realized that one of the major things, if not the major things that we have to overcome is actually the foreign body response. Because trying to do something outside of the body is just going to be a really, really, really difficult putt. It might not happen for 20, 30 years. But if you put something inside the body, that's a good starting point. But every time you put something inside the body, it's like a splinter. So if you could overcome the foreign body response, then you have something. And the approach then is, okay, what can be inside the body that is as biobenign as possible? What's a biobenign material? And then two, are there areas of science that we could actually think about where a foreign material inside the body actually doesn't elicit that foreign body response? It actually encourages it. And that's where the tissue integration comes in. So it turns out that there's a parallel path. Back in the late, I probably started in the 1990s and then into early aughts in the 2000s. There was the area of stem cells was really hot, right? So you remember the whole stem cells, everything was all around stem cells. And one of the notion was the stem cells could actually grow organs that you could transplant back into yourself. And so there were multiple approaches. And one of the more popular approach was you take an organ and then you decellularize it, basically get rid of all the cells. And what you have left is the extracellular matrix. That's a scaffold to make a heart look like a heart, kidney look like a kidney, a liver look like a liver. And that scaffolding encourages cellular growth. That scaffolding does not elicit a foreign body response. Now, it's your scaffolding, of course, so it's not going to do that. But people have tried kind of artificial scaffoldings to see if there's a way. And the intuition or inspiration then is the extracellular matrix scaffolding is trying to do exactly what we want to do. And that exactly what we want to do is encourage healthy tissue to not grow in one layer, but growing a three-dimensional layer, and direct that healthy tissue growth in a way. And you have three-dimensional layer in a sensor, then you have much more surface area to actually take the measurement. And so this whole tissue integrated notion of creating a hydrogel with some space inside that allows tissue to actually go through it healthy tissue go through it, and anchor the sensor there, was our hypothesis of overcoming the effect of the foreign body response in conjunction with a biobenign material. So that was the approach, and we went to work at it. Once you actually solve that problem, and there were a lot of problems actually need to be solved, then the next question is, okay, then how do you get the signal out? Because in order to not elicit a foreign body response, you can't introduce things in the body that's hard, because it activates stretch receptors on the cells, and that's one of the ways that started the cascade. And so in this hydrogel sensor that we inject under the skin, there are zero electronics. There are no batteries, coils, antenna. It's just a hydrogel material constructed a certain way with the chemistry that's inside. And so if you can't have direct communication of signal and you don't have antenna to go out, then how do you actually transduce light? And the solution is via light. So you do that by light, and tune the light in a range that will go through the skin, but won't hurt DNA or cells, right? So it's a near infrared range. If it's invisible range, the light won't go through. If it's in the ultraviolet range, it'll go through, but then it'll damage DNA. But in near infrared range, it goes through the tissue, doesn't do any damage to the cells. And so we created fluorescent moieties in that range. And then the rest of it is a little easier, right? Then you go, okay, then how do you read it on the surface? Excitation, light, photodetector, gather the raw data, and then you start getting to work on algorithms to correct for the raw data and correct for signals and variability and such. I know I talked a long time about this, but I agree with you. We like to think that the solution is elegant. There's a layer of complexity behind it that took us a while to figure out, but I think the fundamental approach is really pretty sound.
Yeah. Yeah, no, clearly. I mean, and again, I personally appreciate the whole, interstitial spaces, extracellular matrix dynamic. I mean, we focus so much on the show on the, cell, tissue, and organ, but we forget about sort of all that surface area that, the other part of the body, and that's where a lot of stuff happens. And I think it's not just an elegant approach, but it fits so nicely into some of the commercial or close to commercial opportunities that you're working on there at the company. And I'd love to spend a little time. We'll talk about glucose in a bit, but let's talk a little bit about the oxygen platform, because here, I mean, this is not blood. I mean, so a lot of us obviously had a blood oxygen thing sitting around since COVID happened. And, okay, this is see how I'm breathing and all that bit. This is oxygenation of the tissues themselves, which when you really dive into it, obviously oxygen extremely important for life, but we're talking peripheral artery disease, wound healing, all the cancer situations where tumor hypoxia is an issue, sepsis, heart failure. I mean, it is no joke. We forget how important oxygen is and tissue oxygenation, but we don't have a tool for it. And now you're bringing it to us. Again, take a little time with this one, because I think it's very, very important and very timely.
Yeah, I'll start again. We build off the shoulders of giants, and we love the innovation that happens in this space. So when we were developing the sensor, one of the things that we figured out, or at least the watch out for, if you will, for the way our sensor is actually made, is because we don't have a tether like a needle through the skin. What happens is, and because our sensing element is separated from the reading element, and there's a small space of tissue between it, right? The light goes through. What potentially can happen is if you have a reader on your arm and you take a measurement and you roll over and you sleep on it, your sensor signal could be declining, but you don't know if it's declining while you're laying on it, because the molecule you're measuring it is actually declining, or is because you laid on it and you compress the space and not enough of what you're measuring is actually getting to that local area. And so our approach to solve that is, well, we should have a reference molecule. And if the reference molecule is actually moving up and down, then it'll tell you what's going on, right? If the reference molecule is staying the same, then you know it's not because of any kind of compression or any kind of restriction. And it must be, the signal decline must be because of the biochemistry you're looking at. So Uh, to your point, oxygen is everywhere. Oxygen is in abundance, and oxygen is so important. Oxygen should be our reference molecule. It's not an obscure hormone or whatever. So we started to get smart and try to get smart about oxygenation, tissue oxygenation. And the smartest people around for tissue oxygenation are physiologists and vascular surgeons and surgeons that deal with wounds. And every one of those surgeons that we spoke with said, "Oh, we understand what you're doing. Okay, here's a little lesson. By the way, you guys should not give up on the idea of the tissue oxygen sensor being a product." You say, "Why?" "Because there's no good way of doing it today." And they explain the problem to us. And that's actually what inspired us to say, okay, let's see if there's tissue oxygen is a problem. And it turns out it's a huge clinical need, and there's a huge demand. Why? To your point earlier, most people think oxygen measurement through COVID is measured through like the clamshells on your fingertips that you see, or through an Apple Watch. And all of those measurements are, those devices are great, but what they measure is how much oxygenation is in your arteries. It's why it's called pulse oximetry. It requires pulse for the measurement to actually be taken. And it basically take advantages of, the fact that we know since we're a kid that, arterial blood is red because it's got oxygen, and venous blood is blue because it doesn't have oxygen. There is a significant colorimetric and spectroscopic difference between oxygenated blood versus deoxygenated blood. And so it's easy to actually measure the degree of oxygen saturation inside the blood vessel. What we measure, because the sensors in the interstitial space, is what we measure is not in the blood vessel. What we measure is actually what the cells actually experience in the interstitial fluid. So you could have oxygen in the blood vessel, but the oxygen may not get to the cells for a variety of reasons. So pulse oximetry would tell you you're doing fine, and the cells may be starving of oxygen. And the crude example that I use here is if your audience have a automated sprinkler system for a lawn, what pulse oximetry measures is whether the pipes have water in it. What we measure is whether the grass is actually getting wet, whether that patch of dirt is actually getting the water that the pipe is bringing. And that's such a significant clinical difference for tissue perfusion, for wound care, for organ resections, for cancer surgery, for COPD, because what's important in many of these measurements and what's the clinical need that's not being met is, am I going to heal this wound and have the outcome that I want in the case of COPD and peripheral arterial disease? Not COPD, I'm sorry, CLI, critical limb ischemia, or peripheral arterial disease. Is this wound going to heal? Because I'm a vascular surgeon and I'm opening up vessels to restore blood flow. I could have technical success and be very happy with my technical outcome. And all these guys are incredibly technically competent, but still not have the confidence that the wound's going to heal, because I know I just restored water in the pipe, but I'm really not sure if that water is going to get to that patch of grass that's actually dying. And our sensors in the tissue, for the first time, could tell the physician that in real time, and even potentially in post-surgical follow-up, because our sensors overcome the foreign body response and they last for 6 months at a time, as opposed to a couple of weeks.
You mentioned the the continuous glucose monitoring advancements, obviously, that this offers as well. And then it's interesting. I looked, was looking back at some of the previous work. I know several years ago you guys had a DARPA grant, again looking at some of the, like the acute stuff in the field, and I've done a bunch of episodes with Jeff Ling and Lenny Tender and Tom Boothby, who was involved in the DARPA Golden Hour project. But clearly, we're talking about some of the stuff that we want to monitor long term in the kind of the chronic diseases, but a lot of this military stuff is it's short term, right? We got to make sure our soldiers are the chemistry is right, and if there's an injury, what's going on and all that. Can you talk a little bit about some of the longer term targets? I know things like lactate and urea and all sorts of other stuff are on the list there, but and if anything is confidential or is part of it, we're going to talk. But take us down that path as well, if you would, because I think it's an important part of the Huntley history.
Yeah, I mean, we're two new friends talking, right? So I think it's technologists talking about what if and what's possible.
Right.
We're really proud of the DARPA relationship we had. Our company has progressed beyond the technology readiness level of DARPA funding, but through early parts, we received a large portion of funding from DARPA over a period of probably 7-8 years or so. So it's not one check, right? It's
Sure.
the first funding was a very modest amount, and they progressively get more and more as we hit our milestone. And DARPA's interest, and it's been a pleasure to work with DARPA, and we're really proud of our relationship with them. And we're proud because DARPA is so good at vetting technology approaches. I mean, that's kind of what they do. They see everything, and for them to actually say, "Yeah, you guys are worth betting on, and here's our support," was very meaningful to us. And they, without speaking for them, our guess is they're thinking about this in along a couple of vectors, right? One, in just general training and soldier health, if you can monitor a soldier's biochemistry, whether it's in training or whether it's in battle, and be able to detect stress earlier or optimize and push the body to the appropriate optimal limit for faster ramp up of the results that they actually want, I think that's an incredibly valuable tool. And tragically, our brave men and women in the military, a lot of times when they go through training, they push themselves so hard. Every so often you hear about soldiers losing their lives because they push themselves so hard and they have a stroke or have a heat stroke and never come back. And so, there's this rightful concern about training and optimization of soldiers' health. I think there's a second part that they want to actually have is if you can monitor somebody's biochemistry, specifically a marker like lactate, then trauma resuscitation, looking at continuous lactate measurement is really, really important in the short term. And it doesn't take a whole lot of imagination to understand why DARPA and the military would be really interested in that, right? Like, how do we better apply therapeutic or resuscitation measures for a trauma patient when they're injured on a battlefield? And having a lactate sensor that's always there, without the need to carry expensive equipment and such, that you could be deployed just right there with the soldier, I think is really important for DARPA. And then the third area that they actually I think that they would be thinking about, and our sensor would potentially be very applicable there, is around the unknown. So if you can monitor certain biochemistries, you understand normal versus some infected state without understanding pathogen. So, and I think I understand the concept a little bit, right? In that I think we all understand the concept a little bit, and we all had experience of saying, "Hey, honey, I don't feel so good." Well, what's wrong? Do you have a fever? No, I don't have a fever. I don't have a cough. I don't have any symptoms, but I just feel like I'm getting sick. Yeah. And then, sure enough, 2 days later, you're sick, right? And the symptoms coming, I got a 100 fever and such. But I don't. For the most part, we kind of always feel like, hey, something is a little off, even before symptoms materialize and the symptoms happening. So that there are sympathetic and parasympathetic and central systems that react to systemic infections before actual symptoms, mass, fever, these mass symptoms, actually fully blown symptoms come aboard. But there's some cascading events that are being triggered inside the body that you could feel, which most likely could be linked to some biomarker somewhere. It could be something as simple as oxygen, right? If we measure oxygen in the interstitial fluid and all of a sudden there is vasoconstriction, systemic vasoconstriction, because the body is like, hey, there's some attack here with a pathogen. We got to keep all the blood closer to the core, not the periphery. You might be able to pick that out just on tissue level oxygen, as opposed to you can never figure that out with oxygen actually in the vessel. So you can imagine a soldier or civilian, for that matter, get infected with something. You don't know what it is. You don't know what a pathogen is, but you know what the body's response to the pathogen is. And it's a systemic response that's pathogen blind, right? Or pathogen agnostic. Then you just catch that infection first, quarantine that person until you spend the 6, 7, 8, 9 months to figure out, or you could do that before you spend the 6, 7, 8, 9 months to figure out what actually a pathogen is, and then you develop a test, and then you develop a vaccine. And you could imagine that's really important on the battlefield, where a squadron comes back sick, and it could be biochemical, it could be biological, it could be chemical. We don't know what it is. But before the pathogen is actually identified, you could actually say somebody, hey, you're coming down with something, right? Let's keep a closer eye, maybe not have you contact with us, platoon, and we'll find out in a day or two. And then in a day or two, the symptoms appear. Good thing you didn't infect everybody else, because it's how pandemics actually spread, right? Is in the asymptomatic stage in which you spread the virus around. So those are, my guess, are the three things that continuous monitoring is actually helpful in that area, and in our relationship with DARPA. And by the way, I love talking about this because it actually goes back to some of my original point, right? Listen, we at Profusa have a great plan for the first product and what we're going to do as a company to execute. But similar to the story that I told earlier about the automated DNA sequencer, I think the potential to measure biochemistry in real time in the interstitial fluid, in a compartment that physicians and the healthcare community understand what the compartment does, but there's not a lot of measurement being done in that compartment, which really is the business end, if you will, of why compounds or why nutrients are actually delivered by the circulatory system and lymphatic system. The business end is in the interstitial fluid. If you can measure real-time interstitial fluid biochemistry, multiple parameters around variety of different indications of population, I think there's just so much more knowledge and insight and prevention that could actually occur, that I think that's a future that we should be very excited about.
Yeah, I completely agree, and I love the preparedness angle there, because that fits like beautifully. Again, we profiled the folks at DITRA and Ian Watson at the Pentagon, actually, and this whole move towards, hey, we don't care if it's Ebola or Marburg or whatever, but if we can find out, as you were saying, those early stages of what's happening, we can shut those down. The whole countermeasures area, it becomes a lot easier. And yeah, no, it's really exciting. And just a side note on that. But what's going on? I mean, I know you have the CE mark per the European Union. I know it's still investigational stage per FDA, but walk us through a little bit of what happens for the regulatory side of things on both, well, the oxygenation, but also the continuous glucose work, and where we sit sort of in 2026.
Yeah, so I want to make sure that we're clear on the oxygen sensor. We currently do not have the CE mark. So this is a little bit of a we hope to get it soon, but we don't have it yet. And this is a little bit of a lesson on entrepreneurialism, right? We're a small startup company. We're a small company, big ambitions, good support. We received a CE mark on the oxygen sensor platform right before COVID, and then once COVID happened, we did a launch. We were selling the product, and COVID happened, and we lost access to the hospital, like everybody else did. I'm not complaining; it just happened. Everybody lost access to the physicians in the hospitals, so we couldn't actually go and train physicians up. That lingered for a while. The investment community and deployment of funding dried up because the world was so uncertain at the time. We all recall. And so that put us in a really tough spot. So our operation was basically paused for a while. Our CE mark lapsed. We went public July of last year to get funding back into the company, and now we're back at it strong. And so, as I mentioned, we are updating our files and regaining our CE mark very, very soon. And so that's where we are, right? We are basically in a restart mode for the oxygen sensor. We are excited of the support that we actually receive, even before the CE mark is actually received, through our clinical studies and the PIs that actually, principal investigators that ran our clinical studies with us in Europe. Their enthusiasm for the technology have not waned. We recently announced a partnership with the Mayo Clinic, for example. They reached out to us, and their enthusiasm about our technology is incredibly palpable. it's taking our sensor technology, oxygen sensor technology, and are there applications in the surgical suite that could be readily applied to. And so the oxygen sensor should be on the market very, very soon as we regain our CE mark, hopefully over the next couple of months or so. And then the next job is to get that over to the US for FDA approval. We had early studies in the US already at UCSF and UCSF and the VA hospital in San Francisco. And we'll continue to push that program along, work alongside the Mayo Clinic as well with with the physicians there to actually accelerate that process with the Mayo Clinic and our work. So oxygen sensor is our first platform. It's for critical ischemia, peripheral arterial disease. This is beginning to sound like a commercial. I don't mean it to be IR, but it is a very important, I think, first step for us to be able to take this platform and be able to demonstrate that, hey, this actually works in a very critical component. Because I think when you do that in a critical patient population, a high value patient population, then I think spreading it out becomes a lot easier. We want to set the bar high and then be able to apply it elsewhere.
And just one other thing I just want to ask you about while I have you today, and I really do appreciate your time, looking out over the horizon a little bit. Clearly a very hot topic for us the last couple of years has been sort of this broad term of longevity or health span. We hear a lot about biohacking nowadays, and actually when I had Lee on the show, the main thing he was talking about are these biosensing for these multidimensional aging models, whether we're measuring mitochondria health or this factor over here or whatever the biomarkers may be. But this is clearly becoming a very interesting area, and continually monitoring what's going on that front is extremely important. You got folks with CG, continuous glucose monitors and this and that of the traditional type, but where are we going? Look over the horizon a little bit. Take us into the future of some of this biohacking stuff with your technology. Actually.
Yeah, it's such an interesting area, right? And I think about it every so often, and I go in sprints thinking about this. And listen, I'll do a little commercial by myself. One way that we could all be immortal is make an impact on society, and people will remember you. Make an impact on your family, and my kids will remember me. And through the stories that my kids tell my grandchildren, they'll remember me too. So I work pretty hard on that part to make sure that I'm immortal through what impact that I could actually have in a positive way. But the second one is obviously the mechanical of, right, how do you understand? But as with business, as with academics and study, as with kind of anything that you actually that we actually do, the ability to actually measure a parameter is critical for understanding what your success is going to be in the outcome that you actually desire. And I think our sensor platform potentially could play a really great role in that, right? That you can't have efficient deployment, if you will. It's very difficult for whatever it is that you figure out in longevity, mitochondrial health, for aging, telomerase, transcription, on and on. I'm not an expert in that space, but there are a lot of parameters people understand what causes aging on a cellular level, and then see if we can actually kind of reverse it, et cetera. Being able to measure something is going to be a critical component for it to be broadly deployable, because retrospective measurement and doing these tests and such for the early adopters is meaningful, especially in the research stage. But if you actually want to get it out there, you need something that's actually easy to deploy. And that's something that's easy to deploy has to have an element, in my mind, inside the body. Once again, most of those things that you're actually talking about can't be measured through the skin, right? Or optically, and trying to magically fire IR signals through, or some other signal through, and trying to get a signature out. And so you got to put a little something inside the skin, inside the body. And when you put something inside the body, I think fundamentally our modality, or some flavor of our modality and what we actually have developed would eventually play a pretty central role in that. And it might actually be that, the impact of some of the activities that's required for these longevity prescriptions, if you will, ought to change biochemistry in real time in a way that's measurable. So, if you um, go down the path of these longevity clinics, it ought to mean that some of the basic things that we measure in an outcome becomes more efficient, right? Or is more efficient than you would be for your age. So your ability to process glucose, your ability to recover from oxygen deprivation, your lactic acid buildup, how efficient your cell is actually using oxygen and other parameters, how efficient it is actually excreting waste. Those things ought to be measured. They might be a proxy measurement. You might not be able to measure directly your mitochondrial health, but you ought to be able to measure some chemistry, because your mitochondria being healthier ought to have an impact on the chemistry. I think that's where we actually come in.
Well, it's going to be an exciting future for your company, I got to say. It's really impressive what you put together. I love seeing, these things get towards the market and the fashion that you're bringing them, and starting out, as you said, with great science. But it's really impressive work. I hope we can do a follow-up down the road to see how all this progresses. But again, really impressive. For everybody out there that is going to be listening to this episode, across the various podcast networks, or who will be watching on the YouTube channel. Again, you've been spending time with Dr. Ben Hwang, Chairman, Chief Executive Officer of Profusa, doing really amazing work, as they say, transform both personal and societal health through these real-time molecular insight tools. Ben, again, thank you for taking the time out of your schedule to join us today and educate us on a lot of really interesting topics. Thank you for everything you do there for human health. And as we like to say here on our show, thanks for helping to create a better tomorrow through what you're developing. Really an awesome story.
Ira, thank you so much for having me. I hope you and your audience could tell I love talking about what we do. It's a passion of mine, and so I never mind talking about it. I don't mind talking about it. I appreciate your willing to listen. I appreciate your audience for turning in.
We do a great job of it. It was great having you.
Thanks.